Mr. Chang and fellow students. We are Group 4 Science Works 1 to 3, and today we will be presenting to you our science experiment. I'm Ying Ting, the group leader, and these are my fellow group mates, Rui-En and Jermaine. Our topic for our science project is biology, and we'd like to particularly study about the effects of the exposure of UV light on bacterial growth. Our aim of the experiment is to find out if the longer amount of time exposed to UV light will result in decreased growth of bacteria. We knew through prior knowledge and some research that bacterial growth is affected by factors, namely temperature, humidity, and light. Our main focus of today, however, will be on the factor of light, because UV light is one of the components of the light that comes from the sun. It's also known that UV light prevents the growth of bacteria by mutating the cell. Thus, based on this knowledge, we hypothesized that the LB agar plate that is exposed to the longest duration of UV light, which is 9 minutes, we have the least amount of bacterial colonies, followed by 6, 3, and 0 minutes. We will then conduct an experiment to prove our statement's validity. Now, I'll hand over to Jermaine, who will be explaining the procedures of the experiment.
Hi, I'm Jermaine, and we will be moving on to the procedures to carry out the experiment. Firstly, the apparatus and materials needed are 12 LB agar plates, a hood, 10 ml of E. coli of 1011 CFU per ml, an incubator, and a stopwatch. You would see some of the apparatus used in the pictures in the videos provided later. We also need paraffin for ensuring the samples are tightly sealed, PS membrane for filtration, a permanent marker, gloves, masking tape, 70% ethanol solution for disinfecting the area, and a long-sleeved shirt. These are the procedures done to expose the LB agar dishes under ultraviolet light. First, put on the gloves and long-sleeved shirt or jacket. This will prevent contamination of the samples by bacteria or dead skin cells from our exposed skin. This precaution also prevents us from being exposed to some of the harmful ultraviolet radiation, as excessive amounts of such radiation can be harmful to human skin. Secondly, turn on the hood and switch on the light and the blower built inside the hood. This is the step of the sterilizing process of the hood to prevent any bacterial contamination by blowing out any floating microscopic organisms. After 5 minutes, spray 70% ethanol solution on the surface of the hood and wipe it with a clean cloth. Ethanol solution helps to disinfect the tabletop where we'll be doing our experimental setup. In order to ensure that the environment is completely sterilized of microbes, close the hood and turn on the UV light for 10 minutes for the final stage of sterilization. Now we can finally begin our experiments in a conducive environment. Using a stopwatch, put in 3 LB agar dishes for each timing and remove after 9 minutes, 6 minutes, and 3 minutes respectively. Package all the Petri dishes using parafilm to prevent the dishes from opening up accidentally and being exposed to the air. Incubate the bacteria overnight in an incubator. The optimum temperature for E. coli to develop is 37 degrees Celsius. E. coli bacteria is used on our experiment, and it is a relatively simple model to develop our results on. Now I'll pass the time over to Rui-En, who will continue explaining the experimental procedures.
Thank you, Jermaine. Now, I will explain the rest of the procedures. Moving on, the procedures to count the colonies of the E. coli growing on the LB agar after incubation. As it is impossible to individually count the colonies of E. coli, we decided to do an estimation. We took an area and estimated the number of E. coli bacteria growing on it. We multiplied by 4 as the area that we took was 1/4 of the Petri dish. Then, we entered the results into a table, including the control, which were the dishes exposed to 0 minutes of ultraviolet light. We also calculated the standard deviation, which will be shown later. Now, we will show you a video of the apparatus required. These are some of how the apparatus look like. These are the gloves that we use to cover our hands, and the long sleeve jacket that we use to conduct experiments. This is the 70% ethanol solution and the incubator hood which we disinfected. These are the Petri dishes. And this is the PS membrane filter that we had used to filter off any unwanted contaminants. And this is also another picture of the UV hood. And this is a picture of the parafilm. And here is another video on some pictures taken during the procedure. This video also includes the results at the end of our experiment. This picture shows us preparing the E. coli solution. This is the micropipette that we use to strip the bacteria onto the petri dishes. And we were doing it inside the UV incubator, or UV hood, and when we had disinfected the area already. We had to do this for all 12 petri dishes. This shows the packaging using paraffin of the petri dishes. As you can see, there are some E. coli bacteria colonies growing on the petri dishes. This is the UV hood and the model and some of its information that we used during our experiment. And these are the results of our incubation. As expected, the petri dishes that were exposed to 0 minutes of UV light had the most bacteria growing on it, and those petri dishes exposed to 9 minutes of UV light had the least bacteria growing on it. Now, Yingting will take over to elaborate on the results of the experiment.
The results are that a big agar plate exposed to the shortest amount of UV light, which is 0 minutes, had the most bacterial colonies growing on them. These samples serve as our control setups. The results show the natural growth rate of E. coli without being exposed to UV light. These are some pictures of our LB agar plates. These plates are the ones exposed to 9 minutes of UV light. You can see that there is very, very little bacterial colonies growing on it that can be considered negligible. While these are the LB agar plates that are exposed to 6 minutes of UV light, there's a moderate amount of bacterial colonies growing on them. Hence, we can conclude that the duration of UV light exposure does affect the growth rate of the bacteria and from our data obtained. Now, we're going to continue the experiments and results.
These are the end results of our experiment. You can see that the LB agar plates with 0 minutes of exposure to UV light had a mean average bacterial colonies at 130, having an STD of 62.4, while the one with 3 minutes had an average of 31.3 bacterial colonies, having an STD of 25.5. The plate exposed to 6 minutes of UV light had averaged 133 colonies, with an STD of 1.53 followed by 9 minutes, with an average of 0 bacterial colonies and an STD of 0. This slide shows our results compiled into a line graph. The x-axis is the duration of exposure to the LB agar in terms of minutes, and the y-axis is the mean number of bacterial colonies present on the LB agar plates. It is shown that there is a relatively steady and general decrease of the number of bacterial colonies as the duration of UV light exposure increases. The trend between the number of bacterial colonies and the duration of UV light exposure can be thus concluded as such. Next, we will be moving on to the discussion sector of the presentation. To combat the problem of the sleeves falling out of the gloves, we can improvise by fastening a rubber band over the gloves. This will lower the chance of the end of the sleeve from coming out. The experiment could also have been repeated with smaller time intervals to more closely inspect the steady change of the bacterial growth. It will also enable us to ensure that there are no discrepancies in the relationship observed with our gathered data. We could also reschedule another day to completely redo the entire experiment as well as to prevent human error and ensure that our results were accurate and reliable. We could have also handled human error and ensure that we could also have handled the apparatus and materials with more diligence and care so that LB agar will not be accidentally contaminated, rendering our experiment useless. We are aware that whenever preparing cultures for the samples to grow, sterility is of utmost priority. Moreover, additional secondary research could have also been conducted to fill us up on prior knowledge so as to better conduct the experiment and know what to expect. The knowledge could also help us in bettering certain loopholes in the procedure and further reduce the error we could have made. We could also try to look for online reports that are more recent to ensure the information is reliable. We could set a specific time frame, which for example could be like, the report has to be last updated not more than a year ago. Lastly, gathering more information from our teachers and mentors could also have been a massive help to the experiment. Now, Jermaine will take over and conclude this presentation.
Our conclusion is that our hypothesis, which is that a longer exposure of bacteria to UV light would result in a decreased bacterial growth, which was shown to be correct. Our results hence agree with our literature review, being that UV light is able to mutate the cell and disable it from reproduction. This proves that UV light does help to control the growth rate of bacteria. These are our references.